Sorry, I know Adam always wants me to go a bit sooner to YouTube than I do.
No, it's fine. It's just so I have a chance to get the link out in case people want to join.
Thank you. Yeah, I lose track of time because we're almost at the hour now.
Yeah, a couple of minutes. Okay, I see it. Thanks.
Okay, do we want to get started right on the hour? I always feel bad.
I'm already quite punctual, but you can leave a couple minutes if you want.
Right on the hour. Oops, somebody's got their YouTube.
Yeah, I always feel bad.
Whoops. I always feel bad starting things on time. As a chronically late person, I really like when.
Well, it'll take you 2 minutes just to remind people about questions and stuff, right?
All right, so then I will go ahead and get started.
Or you can wait. I don't mind.
All right, yeah, let's get started. All right, so hi everybody. Welcome to actually the 5th installment of the Cell Migration Seminars series. It's actually amazing to me that we've made it this far and still going strong. So just before we get started, as folks continue to trickle in, just a few quick things. So next week, we're having kind of a special seminar. We're having 3 short talks from trainees, and we're really excited about getting to highlight work from these PhD students and postdocs. And so we hope that you'll all show up and support them. So we're really excited for that format. Regarding the schedule, we're all set up for the next 4 seminars, and then in the next week or 2, we'll be scheduling talks for the rest of July and getting into August. So if you've considered submitting an abstract, but you haven't yet, please go ahead and do so. We'll be reading everything and trying to put together the best schedule we can. And thanks to everybody who's submitted abstracts already. And then a quick reminder for how to ask questions. So these are going to be reserved for the ends of the talks. So you can type your question into the chat window, either in Zoom or in YouTube, or you can type the word question or just the letter Q if you can't type the word question, and then we'll call on you. And so we actually really encourage you to do this, ask questions yourselves to make the whole thing more interactive, but both methods are great. Okay, so moving on to today's seminar. Again, we're going to have 2 speakers. We're really delighted to be able to have two folks and highlight both of their work. So first up, we're going to have a shorter talk by PhD student David Brückner, who's studying theoretical biophysics at LMU Munich. So his research focuses on discovering simple theoretical descriptions of complex environmental systems, ranging from cell migration in structured environments to collective. So David, take it away.
Thank you. Okay, thanks a lot for the nice introduction, and for especially also for the opportunity to speak here today. So today I would like to talk about some recent work on confined cell migration we've been doing together with the experimental biophysics group of Joachim Rädler in the LMU Munich. A key feature of many of the processes that we're interested in is that cells migrate through complex and confining microenvironments, such as this immune cell here making its way through this collagen extracellular matrix. The feature I would like to highlight here is how the cell has the physical environment. Similarly, in many of the processes with one another, such as the cancer cells in this microtumor here. Simple dynamical law. But the nice thing about these micropatterns is that they allow us to monitor hundreds of cells migrating through identical environments in parallel. And this gives us access to a huge amount of statistics in the form of the trajectories of the cell nuclei as a function of time. And what we're trying to do is to essentially find a simple description for the dynamics exhibited by these trajectories. And what we're drawing inspiration from here is the long history of modeling in unconfined 2D cell migration, where cells migrate across an unstructured 2D surface and leave such pretty random tracks behind them. And people have found that the dynamics of the system and the statistics of these trajectories can be described by something called a persistent random walk. And such a persistent random walk can actually be written as an equation of motion for the cell. And what this means is that if we know the velocity v of the cell, we can actually predict its acceleration dv by dt if we know the persistence time of the cell, which is essentially for how long the cell tends to go in a straight line. And of course, we have to add some random noise to this to get the variability that we see in experiments. And what we were aiming for in this project was to essentially generalize this framework to the problem of confined cell migration. So again, we have trajectories of the cells, and we're aiming to describe these trajectories by an equation of motion for the cell, which describes the acceleration of the cell as a sum of a deterministic term, f of x and v, which essentially is the predictable part of the motion. So it's what part of the acceleration can we predict, given that we know that the cell is at a particular position x in the micropattern and is moving at some velocity v. And then again, there is some random noise added to this. But of course, there's no a priori physical theory from which we could derive what the acceleration of the cell is in such a complex microenvironment. So we actually use the experimental data to constrain our theory. So we have the positions of the cell nuclei as a function of time in the form of these trajectories, from which we can measure the velocities of the cell and also their accelerations. And knowing all of these things, we can then actually infer what this term f of x and v is given our experimental data. And so to do that, we essentially perform a conditional average on the acceleration of the cell, where we measure the average acceleration of the cell at a position x and a velocity v. And this then gives us this 2D color map here, where the color is telling us the magnitude of the average acceleration of the cell. And we'll talk about interpreting this color map in just a minute. But before we do that, we actually have to test whether this model can really describe the dynamics that we're observing in the experiment. Because postulating this equation of motion here is really making a big assumption, right? Because this is a very simple way of describing what is actually an extremely complicated system. You could imagine much more complicated frameworks required to describe this motion, such as time-dependent terms or some more complex memory processes. So to test if this description actually describes the dynamics, we can simulate stochastic trajectories of the system, which perform similar hopping behavior as we see in the experiments. But importantly, these simulated trajectories actually predict the right quantitative statistics that we observe in experiments. And we didn't use these statistics to constrain the model. So that for the model constraining, we really used short timescale statistics, such as the velocities and the accelerations of the cell. But we're able to predict long timescale statistics, like the time it takes the cell to make a transition. And similarly, the model also predicts the probability distributions of positions and velocities. So having shown that our model can capture the dynamics of the system, we actually want to use it to teach us something about confined cell migration. So our initial intuition when looking at the system was that the cell would be repelled by this obstacle placed by this bridge, right? The cell has to physically deform in order to overcome this thin constriction. And so we thought that it might be repelled and this might act as some kind of energy barrier. But we can now actually use the map that we measured from experiments to test this description. And to make this a little bit more intuitive, we can turn this color map into a flow field where these arrows essentially tell us where the system will evolve, given that we're at some particular position in the micropattern. And what I've done here is to color all of the positions where the cell is deterministically accelerating. So it's speeding up in orange and the other positions in blue. And what you see is that as the cell is making its way towards a thin constriction, it's actually speeding up. So it seems to rather be attracted by this constriction rather than repelled by it. And therefore, we have some kind of amplification of cell velocity here. And this is actually really in contradiction to our initial intuition, where we thought the cell would be repelled by this. And this flow field actually predicts that the cell performs oscillations on this micropattern. So deterministically, the cell would always go back and forth, plus some random noise added to this, rather than staying on one island and then noise driving it over to make this transition. So this provides us with a simple way to think about the dynamics of single cells migrating through these confining environments. But we were wondering whether we can actually generalize that also to two cells interacting with each other. And so this two-state micropattern actually gives us a very nice assay to look at cellular collisions, because the cells are over and over making these transitions and then colliding with each other. And the question that we're asking here is whether we can similarly learn dynamical laws that describe cell-cell interactions. So we performed experiments with pairs of both cancerous and non-cancerous breast tissue cells, and then again collected these coupled trajectories of the cell nuclei. And in these experiments, we see a bunch of interesting events, such as these reversal events where cells collide and then retract and move away from each other, like in contact inhibition of locomotion. But we also see sliding events where the cells actually very efficiently move past each other. A third behavior we sometimes observe are these following events where one cell follows the other. And this kind of analysis might also remind you of the work that Brian has shown in the seminar last week. But what's interesting is that for the two cell types that we have here, we actually observe very different interactions. So for the non-cancerous 10A cells, we find that they primarily reverse upon collision, whereas these cancerous 231 cells are very efficient at sliding past each other and therefore also make these transition much faster. But the question that arises now is whether there is somehow a general framework that can describe both of these distinct behaviors in a coherent fashion. And the way we're approaching this problem is to generalize the description that we had for single cells. So we again write an equation of motion for the cells, which we assume to depend on a term that describes how the cell interacts with its microenvironment, similar to the single cell experiments. And so we're hoping that there is some confinement term that looks similar to what we found in the previous single cell experiments, and that the interaction between the cells might be described by a separate term that would depend on the relative position and the relative velocities of the cell. And that could, for example, take the form of some kind of soft repulsion. So I'm just drawing an example here. And if we perform this inference on our experiments with two cells colliding with each other, we actually find that the inferred confinement term looks qualitatively very similar to the single cell term. However, if we infer the interaction term, it actually looks like something very different to a simple soft repulsion. And so specifically, this interaction seems to depend mostly on the sign of the relative velocity of the cells, whereas a soft repulsion would depend on the sign of the relative position. So the interaction we infer actually looks more like a frictional interaction, which would depend on the relative velocities of two particles sliding past each other. But what we see here is actually seems to have the opposite sign to this friction. So it seems to have some kind of anti-friction behavior where when the cells slide past each other, they actually speed up and move away from each other. In contrast, for the non-cancerous MCF10A cells, we find that the interaction is mostly determined by the sign of delta X, which looks much more like the soft repulsion behavior. And again, we now have to test whether the simple description that we postulated and inferred here actually describes our experiments. And so to do that, we again simulate stochastic trajectories and evaluate their statistics and actually find that they have very similar features to what we see in experiments. And so to summarize, we showed that interacting cell pairs can be described by a separate confinement and interaction component, and that this interaction component can be decomposed into a cohesive component, which could be either attraction or repulsion, and a frictional component, which can range from friction to anti-friction. And we found that the two cell types that we investigated in this work actually lie in opposite quadrants of this interaction space, where the 231 cells have primarily anti-friction behavior, and if you look closely, also some attraction, and the 10A cells primarily exhibit repulsive behavior. And so this gives a framework to sort of summarize both of these actually very distinct behaviors with a single quantitative framework. And with this, I would like to thank my collaborators, especially Alexandra, who performed all of these experiments in the group of Joachim Redler, and also my former master student, Nico, with whom I did some of the analysis on two cells, and mostly my supervisor, Chase Burdish. And with this, I would like to thank you for your attention, and I'm happy to take questions.
Thanks a lot, David.
Everyone unmute, give a clap.
Thanks.
Then you know, yeah, okay.
You just came in. I just heard you.
Jen, do you want to start?
Yes. We have a question here on Zoom from Ankita Jha, who asks, what's the size of the bridge? And is it physical confinement or micropattern is the region where adhesive the ligands are?
Yeah, thanks. So the dimensions of the bridge are that it's 7 microns wide, although we've actually performed experiments with a huge variety of bridge widths. And we find that the qualitative behavior actually seems to be somewhat conserved as we change the width of the bridge. And yes, we're really looking here at 2-dimensional micropatterns. So there's no sort of 3D walls.
I've got a question from Abdul Mami Kakada.
Hey, yeah, thanks for the nice talk, David. My question was for the form of the force F, the confinement force that you have. Mm-hmm. What is the functional form of it? Do you know, for example, how does it depend on the width of the bridge, or what is the functional form of it?
That's a good question. So I'm just going back to the slide where I'm actually showing it. So we didn't find a very simple sort of function to describe this map, but it has many similar features to the van der Pol oscillator, which also has this oscillation cycle, and which also speeds up as you approach x = 0. And as we change the width of the bridge, if you make the bridge wider, we actually find that this amplification acceleration goes down. So if you have a very wide bridge, there's actually no longer such an amplification. And if you want, I can show a quick example. So here we did some experiments where we don't have this thin constriction. We still see that the cell sort of bounces back and forth. But if you look at the dynamics, it actually looks very different to what we saw in the two-state patterns. And so this amplification response really seems to be a response to this thin constriction here.
Okay, what about the interactions when you combine the two different types of cells?
That's a great question. We haven't actually investigated that, but I would be really interested to also look at heterotypic interactions.
Okay, yeah, that was a question from multiple people, so I think that's a pretty popular idea.
All right, yeah.
Suvam's asking whether, so he says, what other types of micropatterns used to verify the behavior on friction and cohesion? For the two cells, we really focused on this two-state micropattern where we have the cell collider. But we have some preliminary data where we're also looking, say, at the stripe that I just showed, where we don't have this bridge. And the interactions actually don't seem to depend much on the precise shape that we use. So it really seems to be more a property of the cell types rather than of the precise confinement.
Okay, great. So we have Jim Butler asking a very Jim Butler-y question, which is, what biologically determines the residence time for each side of the sample?
That's a very nice question. So we have this paper here in Biophysical Journal where we actually varied the size of the adhesive site. So we looked at varying the area of the squares, but also their shapes. So we looked at triangles and circles and so on. And we found that the cells mostly care about the area of this of the adhesive site. But whether there is a single biological aspect that dictates that behavior, I'm not completely sure.
I'm going to switch over to YouTube for a question. So Brian Camley's asking whether in two-cell dynamics, does your model, which uses velocity as the basis, distinguish between cases where cells are pointed away from another but stalled by adhesion?
Sorry, what was the last part with adhesion?
So like the case of sticking cells. So does your model distinguish between cases where the cells are pointed away from one another but stalled by adhesion?
Okay, so yeah, I guess that this question is pointing towards these following events where we would have head-tail collisions, right? And in our model, the cells don't have an orientation, so we don't have something like a polarization, but the cells have a velocity, right? So in this model, we take into account the velocity with which the cell is moving and the relative velocity between the two cells. And that should then account for the direction in which they're moving.
Okay, Amrinder has two questions. I'm going to ask the first one, give you a chance to answer, and then the second one. So the first question is, at what location on the cell is the speed measured?
That's the cell nucleus. So we're looking at nuclear trajectories here. Yeah. And
the setup assigns apical basal polarity, and does that contribute to the specific outcomes that you observe?
In the two-cell experiments, or? Okay, I guess you don't know either.
I don't know, maybe in the model, and he can unmute and jump in if he wants to. Okay,
yeah, so say if we look at the single cell movies, there is clearly, intricate polarity dynamics going on. So the cell is repolarizing and sort of exploring the pattern and then moving to the other side. But that is essentially modeled by the velocity v that we have in our experiment. So we don't model an explicit polarity vector, although that's something we're looking at. So we're looking at these precise protrusion dynamics. But yeah, we're really describing this as
the velocity with which the cell is moving as a function of time.
So can I follow up with that? Is
Sure.
Very nice talk. My question was more on the apical basal because these are essentially 2D substrates, right? So if you did not have that basal polarity, would you still have the same outcomes of sliding versus the reversal in the same way? How would the outcomes change?
Okay, that's a good question. So I'm not sure actually. So I could imagine that if you look at this in sort of a 3D confinement, right, where the cells could adhere to all sides of the confinement, that would actually change the way the cells interact with one another, right? Because yeah, as you say, right now they're only adhering to the bottom, and that could definitely change that. Yeah, but we haven't done experiments with 2 cells in 3D.
Okay, thanks. So we do have a few more questions, but we don't have time to get to them right now. So David, feel free to answer in the Zoom chat. And of course, everyone's welcome to come onto the Slack channel and continue this discussion.
Yeah,
so now
I'll be there on the Slack. Yeah.
Great. Glad that you're there. Right. So David, if you could stop sharing your screen and we'll have Daria go ahead and share hers. And so first, thanks, David, for that great talk. And then now we have our second talk. We've got Daria Siekhaus. She's a group leader at IST Austria. And as a research scientist, she pioneered the model for tissue invasion using macrophages. And now her group is investigating how cells move within the complex environment of an organism. So we're really excited to have her here now. Daria, take it away.
Thank you so much. Also to you guys for inviting me to give a talk. And I'm not sure why I can't advance to my next slide. Okay, there. So it's great to be paired with a theorist. So we go from a very, very simple system to ours. Our lab is passionate about trying to understand how cells move in a very complicated 3-dimensional environment of an actual living organism, as Jenny said. And in particular, the question that we want to answer is how cells can cross tissue barriers. How can 2 distinct cell types communicate and coordinate their activities to allow this to occur? We think this is a fascinating question because it requires aspects of biophysical shifts, biomechanical changes, cell biological alterations, signaling, and thus has a really meaty basic biology aspects to it. But we also care about it because it's very relevant to normal and abnormal human physiology. So as you sit listening to me right now, poised within your tissues are resident macrophages that can respond to any injury and can also regulate the homeostasis of those tissues. And many of those got there by their ability to infiltrate into that tissue during development. In the adult, the immune system can respond to infections, and many immune cells use the vasculature as a freeway with which to disseminate throughout the body, and that presents then the topological problem that they have to get across the barrier of the vascular wall to respond to the infections. If you have a tumor, again, the immune cells need to get inside that tumor, infiltrate into it in order to kill it off. And if your immune system can't do this very well, then your cancer may spread, moving metastatically into and out of the vasculature and then into surrounding tissues. So this question of how tissue invasion happens is fundamentally exciting and important. And we hope to be able to identify what are particular genetic programs and some biological changes that underlie this, because there are some things known, but there's really not a lot of known for all the systems that I talked about. We want to be able to identify whether there are distinctions in these programs that are happening in the different scenarios developmentally during immune responses and during cancer metastasis. And if there are, then it's very easy to imagine how one could use this to affect disease outcomes. And if they're not, then there's a requirement to be able to modulate in a specific cell type or spatial way. Our lab is very basic. We work with the flies, so we're focusing on this question 1, and we're just beginning to touch onto question 2. And we hope with the community to eventually contribute to being able to address question 3. So the system we use is the fly, as I said, and in particular the fly embryo and macrophages, which are the dominant immune cell type. These are very important for being able to defend against bacteria and viruses. They also play important roles during development in due to their ability to secrete extracellular matrix and influence signaling. And they are important for maintaining tissue homeostasis. we do not study the functions of the immune cells, we study their movement, their ability to move from the early location in the embryo at the anterior mesoderm to their final location at the end of embryogenesis, dispersed uniformly throughout the organism, ready to attack any invader that would come in. And this movement has been determined by many other labs and was shown to be guided by the Krasnow lab's work by particular factors, PVFs, which predetermine most of the routes that they follow. And in my postdoc in Ruth Lyman's lab, I discovered that one particular aspect of their migration, their movement into this region of the embryo, which is actually the tail of the embryo, the end of the embryo that wrapped its way around next to the head during gastrulation, in which I will refer to in this talk as the germ band. So the ability of the immune cells to go from their head into the germ band, this actually occurs through a tissue invasion process. And I'll show you our evidence for that, some of our evidence. Here I'm showing you just a schematic drawing or tracing of confocal images that Aparna Ratish, a postdoc in my lab, did. This is focusing then on this region of the germ band prior to and after macrophage invasion. And you can see prior to invasion, there's this close apposition of an ectodermal layer and a mesodermal layer. Here in EM that Alpo did, you can see that there is no predetermined channel. And it's actually the macrophages that make a space for themselves as they move in between the border of these two tissues. And I'll show this to you in this beautiful movie Alpo made, in which she's labeled the macrophages with a red fluorescent protein and the surrounding tissues with a green membrane fluorescent protein. And here you can see they've moved up to the edge of the germ band and then they push their way in, preparing to deform some of the surrounding tissues. And here I'll play it again. So this is really lovely. We can do beautiful imaging on this. And the fact that this happens at the same time in the same place in every embryo makes this really ideal to be able to conduct genetic screens to identify novel programs that are involved. And so we believe this is a really powerful system because we don't need to introduce a metastatic cancer cell or inject an infection. We can just watch this developmentally programmed process happen and figure out what's required. And so what we've been doing is screens to look for things that are, when absent, cause a particular phenotype in which just this tissue invasion into the germ band is defective compared to the wild type, but the movement along the other non-invasive routes of the embryo are fine. And thus we have sort of a natural internal secondary screen to make sure that what we're picking up are programs that are specific for tissue invasion and not for general migration or general cytoskeletal capacities. So we've looked for what's going on in the surrounding tissues and in the macrophages. And today, because I was told that this should be like a Gordon conference talk, I can tell you about two unpublished stories, which are very close to being submitted. One on the importance of cell division in the surrounding tissues to govern and regulate the capacity of the immune cells to make their way in and infiltrate. And the second on a new genetic program that allows the macrophages to boost their energy capacity to meet the challenge of invasion. But I want to first give you a little background and kind of encourage you guys, if you haven't already, to go read our previous papers. So what we found in prior work that I think is important for the context of what I'll be telling you about is that the ability of the macrophages to make their way into this germ band region requires a change in the state of the surrounding tissues, a decrease in the tissue tension, which is triggered by the conserved inflammatory molecule TNF. This is the work of Aparna Ratish, who's now professor at the University of Warwick. So if you're in England, keep an eye out for her and say hi. The other background I wanted to tell you is that we have found that there is a crucial shift in the capacity of the macrophages that leads to a change in specific aspects of glycosylation on a subset of proteins through the activity of a novel transporter that ultimately leads to the ability of extracellular matrix to be secreted and, we think, cross-linked more effectively by the macrophages. And this, I'm mentioning this also just to highlight that what we are looking at is distinct from the invasion that many of you see in these in vitro ECM cultures. So in our system, matrix metalloproteases are not required. They do not help. If we make mutants of them, the invasion is still fine. And in fact, it seems as if the position of ECM is helpful in that it seems probably to give an anchor point to the immune cells to allow them to pull their way, their back end and their nucleus in through the resistance of the surrounding tissues. So I will talk more about these surrounding tissues and their resistance and how they alter it based on the work of a wonderful postdoc in my lab, Masha Akhmanova, She's a biophysicist by training, but she's fallen in love with microscopy. And what she's found is that the ability of the surrounding cell to divide is really crucial for the invasion into the tissue by the macrophages. She was doing imaging because she wanted to understand more about the cell shapes of the ectoderm in order to make biophysical models. And so I'm going to introduce you to the movies she did. I've shown you up until now a lateral view where the germ band is here, and the macrophages are moving between the yolk sac and the ectoderm, and then coming in along this ecto-meso interface. Masha made some movies in which she was doing a dorsal slice here. So the macrophages are coming in this way, diving underneath the ectoderm, and then coming up here. And when she made these movies, she found that the cells of the ectoderm were dividing right at the time when macrophages were making their way in. I'll play it again. You can see on the other side as well. So this really intrigued her. And so she did this lovely profile of what's going on with cell division for every one of the movies that she made. So she's labeling each cell and its rhythm in a different color. So cell 1 is in this color and cell 2 is in this color. And for each movie she's tracking, is that cell dividing, in which case the line is at this level? Is that cell rounding up, in which case it's in this level? Or is that cell not doing either of those two things? So we here get kind of like an EKG of the division of the cells for each movie, the surrounding ectodermal cells. And here she tracks when the macrophages actually make their way in. And what you can see is that this point, when the macrophages invade, is in every single movie she made, exactly when one or both of the surrounding cells, the ectoderm, are either rounding up or dividing. When she quantitates that, taking the difference in time between either the division of one of these two cells or the rounding of one of these two cells and the entry, and then takes the minimum for either of these two, she sees a complete concordance between either the division or rounding of the surrounding ectodermal cells and macrophage invasion. So this is really intriguing. This argues that there is potentially a link between these two processes. We see that the location of the invasion coincides with the cell division, and we see that the timing correlates. Does this mean that there is a causation? Could it be that macrophages and they're trying to enter could in fact prompt division of the cells in the surroundings? Or could it be that the division of the surroundings somehow opens the door and allows macrophages to come in? So to address this question, we wanted to, of course, manipulate one side of the equation and see what happens on the other. So can we change the division rate or block the division and see what the effect is on macrophages? So we've done that in different means. Masha started with a kind of a very forceful approach, injecting a drug, dinaciclib, which is an inhibitor of CDK kinases and thus really blocks the cell cycle to a great extent when she injects it on into the embryo, far away from where the germ band is, to make sure there's no interference. Here she quantitated the division rate of the ectoderm after the injection of this drug, and you can see that it profoundly decreases their capacity to round up and divide. And when she looks then in the germ band of the embryos in which she injected that, she sees many fewer cells making their way in there. And here the quantification, a really profound decrease in the evasive capacity. Now, you may be thinking, aha, yes, this is a very strong effect, but you are injecting a drug which inhibits both the surrounding tissue division and the macrophage division. Could it be that that's somehow what you're actually seeing? So we don't think that the ability of the drug to inhibit macrophage division is important, because work from my other student, Shamsi and Tanani, in which she had blocked cell division in the macrophages themselves or inhibited it strongly by using RNAi for string, showed that actually this led to an increase in the invasion or the number of macrophages in the germ band region using string, cyclin A, and many other components that would inhibit cell division. So the effect we're seeing with dinaciclib, in which when you make live movies, you can see that there's many fewer of them coming in, we think is not due to an effect on macrophages. And our supposition that this is really, the importance is in the ectoderm for the division is enhanced by the fact that the remaining invasion that happens of the macrophages, shown here in purple, is happening when the surrounding tissue of the ectoderm is actually dividing. And here you can see this rounding cell right next to where this macrophage is sneaking in. Okay, but of course the most elegant way to address this is to inhibit the cell division in this ectoderm genetically only there and see what the effect is. And so Masha has done this, blocking string again with a driver that only affects the ectoderm. And she, again, quantitated to show that this leads to a decrease in cell division occurring there. And she sees a quite strong effect. Around half the number of macrophages that normally make their way into the germ band are present when she counts them in multiple fixed embryos. So cell division is important, is required for allowing invasion to occur, but is it really the gatekeeper? Is it really the thing that sets the invasive capacity? So if we increase cell division, can we actually get more of them in? So this Masha did by blocking down triples with RNAi, and again, seeing an increase in the division amount in the ectoderm. And she did in fact see, surprising to me, an increased amount of macrophages here in the germ band, which she quantitated over here. So for this, from this data in its totality, we really make the relatively strong conclusion that ectodermal cell division is one of the main processes that regulates macrophage entry. So, and this is exciting to us because to our knowledge, this is not something that people have appreciated before as being important for invasion. And we're wondering, is this at all relevant for cancer treatment in which you have division inhibitors, and could there be any effect of cell division on the capacity for immune cells to infiltrate into tumors? And so we are working to try to find collaborators to investigate that aspect. But to step back and to summarize what I told you in the background and in this new unpublished work, there's a crucial role for the surrounding tissues and their regulation by an inflammatory molecule, TNF, to reduce the tension, and TNF actually also influences cell division to allow macrophages to make their way in. But our goal is to have a really comprehensive view of everything that's going on. So we also want to know what's happening inside the macrophages. And for that, I will tell you about a story from Shamsi Imtinani, a grad student in my lab, who has found a completely new genetic pathway that regulates we believe, the metabolism of the macrophage, enhancing its capacity to produce energy to allow it to meet the challenge of this grappling with the surrounding tissues and invading in. And this program seems to be particularly important for the pioneer cells, the initial macrophages that make their way and forge the path for the rest of them falling behind. So when Shamsi joined the lab, she was looking for a gene to call her very own. And she found one which had the pattern that we look for, namely of being expressed in macrophages in the time before their general migration is happening and during the time when they're invading. And so this is present during the time of invasion. And it is, the other criteria she wanted was that it was something that was relatively unknown, that nobody had touched before, and thus could potentially lead to completely new insights. And the gene she focused on, CG905, had never been investigated in any system, in any species, and had intriguing hints to what it might be doing. So it has domains that are associated with chromosome segregation and thus presumably DNA binding in yeast protein. And it has two transcriptional activation domains, some nucleolocalization signals, and nuclear export signal. She also liked this protein because it has two mammalian orthologs, which are 40% identical and which share this general domain structure. And these are, in fact, themselves enriched in immune cells in their expression in vertebrates. So I wouldn't be talking to you about this if it didn't have a tissue invasion effect. So here I'm showing this to you. Here is our embryos in which we're labeling macrophages with red fluorescent protein. You can see in the control, there's a certain amount that make their way into the germ band. In the mutant for this gene, CG905, there's a strong reduction. And when we put the gene back into the macrophages themselves, we see an increase. So here's the quantitation. You can see the mutation causes a fewer germ band invasion, and this is completely rescued by the gene being put back itself. So this makes us conclude this protein, which Shamsi has shown is actually in the nucleus, is required inside the macrophages themselves to allow them to make their way into the germ band. So we always like to know what precisely is happening, which step exactly of their movement in is altered. And so Shamsi made movies using a fluorescent marker for the nuclei in control and in CG905 embryos, mutant embryos. And here I'm showing you successive stills to allow you to see this process. And she quantitated everything that you can imagine and didn't find any defect in the speed between the control and the mutant in the migration up to the germ band. But then when she looked, the time that it took the macrophage to make their way into the germ band, the first one in the CG95 mutant was reduced, or it took longer time for that to occur, and the speed with which they moved inside the germ band was reduced. And here's the actual data for that. It takes around twice as long for the first macrophage to invade into this tissue in the CG95 mutant, and the first and the second cell show a slower speed, but not the subsequent cell. So it really is the pioneer cell which shows a defect and not the ones thereafter, sorry. And again, the migration along other routes is unaffected, for example, along this region here, really arguing that this is a very specific requirement for this gene in the invasion of the germ band tissue. So how could it be doing this process? Well, Shamsi first asked, okay, which of these domains are important? And she made mutants in which one or two of these, or both activation domains are gone, this DNA binding domain is gone, or both of these are gone. And she found that all of these are required to allow CG95 to carry out the invasive function. So if she puts any of these mutant forms back into the macrophages that are mutant for CG95, they are unable to restore the invasive capacity. And based on the fact that these domains are required, you will not be surprised when I tell you that the vertebrate orthologs, which share these domains, are completely capable of substituting for CG95's function. So either of these two mammalian orthologs, when put back into macrophages, can restore the capacity of the macrophages to invade, arguing that invasion that's governed by CG95 and the mechanism by which it acts is conserved from the fly to the vertebrate situation. Okay, so I've talked to you about a conserved nuclear protein, which has an unknown function that is important to enhance pioneer cell invasion. And then, of course, the question is, how is it doing that? It requires these transcriptional activation domains. It requires these DNA binding domains. Could it be, Shamsi reckoned, that is actually involved in transcriptionally upregulating specific downstream players to allow invasion to occur? So she then carried out RNA sequencing on control and on CG95 mutant macrophages that she fact sorted to identify these potential downstream genes if they existed. And she found that indeed there was a set of proteins whose RNAs required CG95 to be expressed at higher levels ranging between 6 and 25 or 32-fold higher. And then she asked, okay, which of these could in themselves actually be a direct downstream target of CG95 by which CG905 allows invasion to occur? She tested this by knocking each of these down and seeing which were required for invasion themselves. And she found that these three, two metabolic enzymes and an RNA helicase, were required for invasion to occur. And I'll show you this data and tell you about their function now. So the first metabolic target gene is one called glyoxylate reductase hydroxypyruvate reductase. And metabolism is very complicated. So these enzymes participate in many, many reactions. And so they participate in amino acid production, in breakdown of hydroxypyruvate, but they also play a role in this, as you'll see in the rest of the talk, which we're focusing on, we think is the one that's the relevant one, in helping to produce pyruvate by, through mass action, getting rid of one of the products of an enzymatic reaction that produces pyruvate. That could, if it remained, enhance the back reaction. So this enzyme is itself required for invasion, as you can see when we do the quantification of the number of macrophages in the germ band. When Shamsi expresses two different RNAis in macrophages themselves, there's a strong reduction in the movement in. The other metabolic target is called lysine ketoglutarate reductase saccharapine dehydrogenase. And this again has many roles that it can play. It's been linked to transcriptional inhibition, lysine catabolism in different ways. But the one we're focusing on is the combination of lysine and alpha ketoglutarate in a pathway that ultimately can produce acetyl-CoA, which is a substituent that can lead to energy production in the mitochondria. This enzyme is required. Two different RNAis, again, show a strong decrease in the number of macrophages that are making their way into the germ band. And again, for all of these proteins that I'm talking about, this is a very specific defect. There is no effect on migration along other routes, along the ventral nerve cord. And there's also, for all of these, no change in the total number of cells of macrophages. So this is really a specific invasion defect. And to really substantiate the opinion that these are downstream players that help CG905 aid the pioneering macrophage in its challenge of negotiating its surroundings, is the fact that if Shamsi puts them back, either one of these two back, in the CG905 mutant background, she can rescue to a large extent their capacity to invade. So here you see the control, here you can see in the CG905 mutant, and here is the embryo in which she's put one of these two enzymes back, and you can see that it restores the capacity for germ band invasion. Okay, so what we know now is that this novel nuclear protein is important for the upregulation of these two metabolic enzymes, which are themselves both required for invasion and which both can, to a large extent, rescue the absence of CG905 if we put them back at higher levels. We also, I told you that there's a requirement for this other protein, an RNA helicase. And here I'll show you the data on its functionality. Knocking it down by RNAi, you see, again, strong decrease in invasion to the germ band. And because this was the strongest defect that Shamsi saw for any of the potential downstream players of CG905, she carried out live imaging to really investigate what its relationship was to the CG905 phenotype. And what she found is it really strongly phenocopies and recapitulates what she saw for the nuclear protein. So the time for the germ band entry is again around twofold longer, and the time for the first and the second cell to make their way into the germ band is again slower, but not the subsequent cells. And again, this RNA helicase can restore the capacity to invade the germ band if he puts it down back in the CG905 mutant, which would make sense if CG905 is required to upregulate porthos levels. Putting it back should be able to fix the problem to a large extent. Okay, so then the story now is this nuclear protein is required to turn on these three components, two metabolic enzymes and porthos, each of which are required for invasion, each of which can to a large extent rescue CG905 defect if put back. But why would the cell do this? Why is a cell deciding to turn on two metabolic enzymes and RNA helicase at the same time to allow invasion to occur? Could this RNA helicase be actually involved in metabolism in some way? And could they be working together to do something? Our hypothesis was that porthos, as an RNA helicase, is recognizing a particular RNA structure in the 5' UTR of RNAs to allow translation to occur. And there's precedent for such ideas. This is why we had such a hypothesis. And to examine this, to see if porthos could really be affecting translation, we collaborated with Elliot Martin and the Rangan group to carry out polysome profiling. And in this, Elliot purified polysomes from either control S2 cells, which is a macrophage-like cell line, or this S2 cells in which porthos has been knocked down by RNAi. And then he examined, okay, on those polysomes, if you compare the RNAs that are on those polysomes and to the, plotted here, to the RNA that's present in the cell, which RNAs are underrepresented and thus being translationally repressed in the absence of porthos. And here are the set of proteins that whose mRNAs fit this category. And when Elliot analyzed them, he noticed that they shared many of them shared a particular property, that they had a certain sequence in the 5' UTR, a polypyrimidine stretch, which is called a top-like motif. And so the hypothesis was that porthos can recognize this top motif and help stimulate translation to allow the production of proteins that contain this motif. And to test this, Shamsi made a reporter, this idea that porthos is really required to increase translation of these top-containing mRNAs. She made a reporter in which this top motif is fused next to the open reading frame for GFP. And then she transfected that, or Yulia Biebel and she transfected that into S2 cells, which were either wild type or which were mutant for this upstream nuclear regulator of the RNA helicase or the helicase themselves. These had been CRISPR-Cas9 treated to reduce the copy number of each of these two genes. And what you can see is that the translation of this reporter is strongly reduced either in the absence of the upstream nuclear regulator or in the absence of the RNA helicase. And to a greater extent than the somewhat small, or the decrease in translation that we see for the control EGFP. And so when we look to see, okay, that which proteins are in the cell that have this 5' UTR top-like motif that are actually on this set of proteins that are translationally repressed, we found, to our delight and surprise, that these proteins with this top motif are enriched for being in the mitochondria, the ones that depend on, for porthos on their translation. Many of them are involved in cristae formation, in mitochondrial import, and we even found a component of the complex 3 and complex 5, the enzymes that are, the proteins which are required for oxidative phosphorylation. And so our hypothesis is that porthos is allowing the translation, or the enhanced translation, of proteins that make the mitochondria as efficient as possible, allowing every single electron and NADH that the cell produces to really be converted into an actual ATP. And that the larger model then is that this nuclear protein of CG905 is inducing a metabolic switch, that the upregulation of this nuclear protein is inducing a metabolic switch in macrophages to prepare them to be able to negotiate the challenging environment that invasion requires. This CG905 is turning on porthos to speed mitochondrial efficiency, while simultaneously upregulating 2 metabolic enzymes that are capable of converting amino acids into substituents, pyruvate and acetyl-CoA, that can feed into the TCA cycle and thus help fuel ATP. And we are trying to test these hypotheses by directly examining the mitochondrial respiration rate in wild type and porthos and CG905 mutant mitochondria, and trying to do metabolomics to see if we're really accurate that amino acid levels of these would be higher in the mutants for these two. And we're very excited about this because this is, to our knowledge, the first time in which anyone has found a program which could allow an upregulation of energy capacity in invading cells. The idea that higher ATP would be present in an invading cell is in the leader cell that's pioneering the invasion is not unprecedented. Claire Reinhardt-King has seen this in her studies of metastatic cancer cells moving in ECM. And we're now proposing this could be potentially a generalized program, which would be used by cells in many different contexts to upregulate the production of ATP. And we're particularly interested in our lab in seeing if this process could be conserved in vertebrate immune cells to allow leader cells to be very effective. So to sum up, I have told you about the intricate and beautiful coordination that exists in the genetic changes that occur in the surrounding tissues and in the macrophages to allow this developmentally programmed invasive process to occur. In the ectoderm, there is a change in tissue tension and an increase in cell division triggered by TNF that primes the environment to allow the macrophages to take advantage of this, sneaking in as the surrounding cell is dividing, fueled by this shift in metabolism that gives them more energy, and anchoring themselves, we believe, on the extracellular matrix, which has been laid down more efficiently due to this shift in glycosylation by a novel transporter. I haven't had time to tell you about 2 other unpublished stories in our lab, in which we found that not only these novel pathways are important, but also ones that have been previously linked to metastatic invasion. FOS and BMP are also important in our systems in macrophages. FOS by enhancing cortical actin resistance to the surrounding tissue pressure, and BMP in mediating leader cell functionality. And thus, we are really excited to think that what we're finding is not just a quirk of Drosophila development, but really that we're finding programs that could be more generally utilized. And we have some evidence for that already in our own lab, in which the transporter that I talked to you about that affects glycosylation seems to be also required for cancer metastasis. And we have been collaborating with Francesca Perry's lab, who has shown that one of our shifts in tissue tension, the TNF trigger, seems to also be important for the formation of microglia. And thus, in future years, we're really excited to see how the work that we've begun will affect the broader in vivo cell migration community. If you enjoyed the story, please follow us on Twitter. the Siekhaus lab. I want to give a big thank you to all the members of the lab, past and present. You've seen the pictures of the people that were doing the work I highlighted. I also want to talk about it to like Jurgi, our lab manager, who has contributed enormously to almost all of these projects and the tools that he's built. Marco, who did the work on the cancer metastasis relevance, which I didn't talk about, Vera, who worked on CFOS, and Steffi, who worked on BMP. I want to thank my funders, my former postdoc advisor, who let me start this crazy project in a lab that was devoted to germ cells, and at IST, the Heisenberg lab that taught us a lot about biophysics, tissue tension, and has been also working on ATP detection and has given us some guidance there. We have a wonderful bioimaging facility, EM facility, and a general support from the Institute. And I really want to highlight our collaborators at SUNY Albany, Elliot Martin and Prashant Rangan, without whom the second story would not be what it is in any way, shape, or form. And the Fly community, which is a wonderful group to be a part of. Thank you very much for your attention, and I'm happy to take any questions you have.
Thanks, Daria, for wonderful.
Thanks very much.
Everybody unmute, give a clap. Great, thanks guys. It's our attempt to make this feel a little bit more like in person. All right, I'm gonna start out with a question from YouTube. Sam Colley says, excellent talk, Daria, agreed. I was wondering if the cell division of the tissue is signaling to the macrophage to invade, or whether it's more of a physical gate allowing the macrophages to pass.
Yeah, that's a very good question, and we really don't know. The preliminary data that we have is, so I guess the first model, you could imagine that maybe the division of the cell would make more surrounding space, which could say let the guidance factors that we know be more prominently sensed by the macrophage to kind of trigger their movement in. A second being that potentially the rounding up of the cell and the decrease ECM attachment that would produce could also open a gate. And so we're testing both hypotheses, and we haven't really explicitly produced data yet to test the first, but we have data for the second, which supports the idea that it's actually more of a gate opening, that the disassembly of contacts between the ectoderm and the underlying ECM layer is what benefits the macrophage in order to be able to invade.
I'm going to combine the first two from Zoom together. So this is from Yelena and Amrinda. So do you think the role of cell division is purely mechanical? Can invasion of macrophages in embryos be blocked with blocked cell division, be rescued by reducing tension somehow? And then the follow-up from that is, do you see increased number of divisions in the presence of macrophages?
So we're trying to test the second question, which is a good one. So given that TNF seems to be triggering cell division, and there is some TNF also produced by the macrophages, I would expect the second question, answer the second question would be yes, but we have not yet managed to get rid of the macrophages entirely genetically and then study the cell division rate. And the first question again, can you tell me what that was?
Do you think the role of cell division is purely mechanical? So can invasion where the cell division is blocked be rescued by reducing tension somehow?
Well, actually, I mean, division increases tension, right? So in some ways, we were a little confused by that because I don't think the cell division role is a regulation of tension, because it actually leads to an increase in tension rather than a decrease in tension that we showed previously. But we have not explicitly tested that. I think it would be hard to, we could try it. Because of our imaging, in which we actually have visualized that what occurs during division is a assembly of vinculin-containing contacts, and that we've really strongly linked that to the division that occurs. We've been focusing on that and doing RNAis for that. But it's an interesting question.
So Gita Jha asks, immune cell infiltration in tumors is often linked to physical barriers provided by soft tumors. Do you think ectoderm is doing the same and cell division provides a way to reduce the barrier?
Exactly, yes. That's our hypothesis, yes.
Okay, next one from Olivier Tardy asking, have you looked at apoptosis in the germ band region as a local driver of proliferation or changes in tissue tension?
Yes, so we know as part of our TNF story, we were actually wondering if it could be triggering apoptosis, because that's one of the roles that it's been linked to in different systems in the fly. And we did not, so Apu did some very nice work on trying to visualize apoptosis and see if it was required for invasion to occur. And we did not see any concordance of where apoptosis occurs and when or where invasion happens. And in fact, when she blocked apoptosis, it didn't reduce the number of invading macrophages. So we don't think that that's a really important cue for this process.
Okay, Dennis says, lovely talk, Daria. Do you know which signal triggers CG9005 expression in macrophages with respect to its role in invasion in the germ band?
Yeah, no, so we don't know. So we're assuming that that's the general components that are triggering macrophage fate, like serpent and GCM, are the ones that turn on. It turns on quite early, around stage 9 or 8. So we think it's part of the initial program. And this is one of the conundrums that we've had in our work, is that we keep finding these components which are generally expressed in all the macrophages, but which have defects only in the early ones or the ones that are invading. And so it seems like the logic of the cell is just to get prepared for everybody to do it, and then to have, and then that's needed, and that's then produced in all the cells, and that's only utilized in some. There's only one pathway that we found, PMP, which is itself only expressed in the leader cells and in a subset of macrophages up to now. But we're probably missing a bunch of components, and we're trying to do single cell sequence now to see if there are other subpopulations that define the leader cell program.
A question from Anna Maria Camo, asking if you were able to distinguish if the role of cell division is specifically about cytokinesis, and whether you've looked with regard to cell-cell adhesions. So do you think a reduction in adhesions could facilitate invasion?
That's exactly what we think, right? So given that we see that both for the rounding and the cytodivision, that the invasion happens either with both of those, I would say that argues that it's not specifically about cytokinesis. And the fact that we see delamination or disassembly of the ECM contacts, and our preliminary data on RNAi reduction of that seems to argue that it can potentially rescue the block in division.
We have another question from YouTube. Ian Veloso Randolph asks, do you think inhibiting cell division in macrophages increases invasion because it somehow increases metabolism?
Potentially, right? Because I don't know if the person is thinking as I am, but TOP is known to be, the TOP sequence in 5' UTRs has been known to be involved in the TOR pathway, and the TOR pathway can be activated when cells are no longer dividing. So it could be, in fact, that is one of the mechanisms by which there's an enhanced energy production. And we would love, we were not able to get ATP sensors to work in vivo to allow us to really check that out. And so all of our attempts to do the metabolism require us to extract the cells out and examine them. So that's a bit harder to do, but we could try to say treat our S2 cells with cell division inhibitors and see if that alters their mitochondrial profile by C-cores.
Question from Juliana, who's asking whether it's the macrophages themselves that are the source of the TNF, and whether you try to knock down TNF to check for the division rate.
So the TNF, we have the work that Apu did was focusing on and has showed that there was a crucial role for TNF being expressed in the amnioserosa, which is the tissue that flanks the germ band. So there's a local tissue-tissue interaction. But Apu has shown also that there is TNF required in the macrophages, and Masha has shown that in the TNF mutant, the cell division rate of the ectoderm is decreased. And we would assume that then that's a decrease due to TNF coming from both the amnioserosa and the macrophage, but we haven't explicitly tested that yet.
A question from Yukata. Mitochondria would need more oxygen to produce more ATP. Do the hemocytes have a mechanism to take in more oxygen for their invasion?
That's a great question. They're kind of, I mean, they're sitting right before they dive into the germ band. They're sitting on top of the germ band underneath the amnioserosa with kind of a big, so it's probably the place in the embryo which is the closest to the outside for the diffusion. So maybe they're kind of tanking up at that point right before they dive in to invade, but it's a fabulous question and we don't know.
Question from Ruth Johnson, who's asking whether you can change the route of the macrophages by precisely controlling where the division occurs.
Yeah, no, we wanted to try to do that. So we were, what we did do is try to just modulate tension, thinking that maybe the signal that the macrophages feel to say, okay, the cell is dividing is the increased tension of the rounded cell locally. And we collaborated with, and I'm blanking on his name now, Sturensis and Embl to try to induce optogenetically the rounding of cells and see if that would then get them to go in, but that was not sufficient. We have not yet found a way to kind of locally optogenetically induce sort of the kind of, tribble or something to stimulate it, but that would be the way to test it. And it's a very interesting question. We would love to be able to try that.
Great. So I think our last question comes from Amrinder, who first says, great talk, and then asks, in your in your cartoon description, you show a long invading protrusion.
Mm-hmm.
One, do you see increased or decreased protrusive behaviors in your studies under different conditions, especially related to energetics? And two, what about leader or follower cells?
Okay, so a lot of our analysis is done, initial imaging is done just with the quantification of the nuclei. So it has not been that we have looked for protrusions in all of our situations. The one in which we did was in CFOS, and the CFOS mutant, we actually got more kind of protrusions happening, which is fitting with what the role that has been known for it in previous contexts. But the other question was about leader follower protrusions.
I think, yeah, do you have, what are the protrusive behaviors in your leader?
Okay, so the interesting aspect about the system is that the cells seem to go in kind of a fluid chain. So they're not a collective, they're not, affixed to each other, but they do display semi-collective behaviors in that as you're imaging it, it looks like the follower cell will kind of produce a protrusion to touch the one ahead of it and then retract it and produce it, touch it again. So it's kind of like a line of elephants with, with a trunk grabbing for the tail and then dropping it and then going to grab it again. But yeah, it's an interesting question about the effect of the energy on the protrusion. And we should maybe prepare to be able to answer that question in case a reviewer asks it. But we have not yet investigated that.
All right, great. Thank you so much. So I think
that was fantastic.
Yeah, that was really
great. Thank you so much for having me be a part of this really exciting venture, and great work on organizing all this.
Thank you. We really appreciate that. All right, and as I mentioned before, we can definitely continue the conversations on Slack. Both David and Daria are both available on Slack for continued question answering. And thanks again. If you guys want to unmute and we can do a quick one last thank you to both of our speakers.
Thanks, Daria. Thanks, David.
Thank you.
Thank you.
Thank you.
Woo! All right, thanks so much.